Hi, my name is Aline from the University of Tübingen in Germany, and my research project is about developing in vitro testing platform on mature pancreatic beta cells in the context of diabetes research. There is already a lot of research that has been done to understand type 1 and type 2 diabetes in human, and most of it has relied on the use of animal model. For example, we can inject STZ into mice, and this will destroy their pancreatic islets and mimic some of the conditions seen in type 1 patient. There is an important ethical concern about the use of animals, and there is also major physiological differences between animal models and human. For example, rodent pancreatic islets will respond very differently to glucose compared to human islets. There is also differences when it comes to the immune system between rodent and human. All of that will contribute to the lack of translation from preclinical trial to clinical trial in human. There is an alternative, is to use materials from human donors. We can isolate Langerhans islets from a human patient and perform in vitro experiment to understand better the physiology of those islets. The problem is that as primary cells, those pancreatic beta cells will lose their function over time. Over about 1 week, they won't respond to glucose anymore. And this is where my projects come in, where we aim to develop in vitro platform for diabetes research using stem cell derived pancreatic beta cells and combining it with the organ on a chip technology. So stem cells have the potential to differentiate into any cell type of the body, including pancreatic beta cells. So this way we are addressing the issue of islet shortage from donors and the use of animal cells, because stem cells derived from human have a human genetic background. On the other hand, we are using the organ on a chip technology to provide a better microphysiological environment to those cells. Another important part of the project is not only to provide a vasculature-like flow to those cells, but also pancreatic niche molecules. In the body, cells are surrounded by extracellular matrix, and this helps them to function and survive over time. So here in this project, we also use different type of extracellular matrix and provide it to our pancreatic islets on that chip to improve their function and survival for long-term studies. The last part of the project is to provide non-invasive tools to monitor our pancreas on a chip platform over time in a non-destructive way. For that, we're interested in 2 different imaging techniques. The first one is Raman microspectroscopy, and the second one is fluorescence lifetime imaging. By combining the different parts of this project, stem cell-derived pancreatic beta cells, vasculature-like flow, pancreatic niche molecules, and non-invasive tools, we can create in vitro platform with a highly physiological relevance for drug testing. And this will ultimately reduce the use of animal model in the research field of diabetes. I am very thankful to have received the IFER fellowship, and I'm looking forward to make my contribution to the field.